thank you very much for that introduction, and thanks everybody for listening to this talk. So, yes, without deviating too much from the main title of the talk, I'm gonna talk about some phylogenomic insights into the taxonomy and biogeography of Weinmannia. And yeah, as Alex said, I just mentioned that what I'm going to present here will be part of my PhD dissertation as a PhD student at WashU. And yes, broadly speaking, the aim of my thesis is to contribute in the understanding of plant biogeography and the ecoevolutionary processes that shape the diverse flora in the Andes and the Neotropics in general. And for this purpose, I plan to use Weinmannia as our study system. And today I'm going to talk about two of the objectives I have in my dissertation. And the first one is to estimate the phylogeny of Weinmannia while accounting for integration and hybridization. And the second is a bit of the generate some understanding of the biogeography of the neotropical Weinmannia. And well, to start with, Weinmannia is a diverse genus of trees and shrubs. They are mainly distributed in the Neotropics with around 75 to 90 species, but we can also find two species in the Mascarene Islands here. So the genus is characterized by having these small fragrant flowers that are likely to attract bees and other small insects. And they also have these hairy and dust-like seeds that are easily dispersed by wind. So the genus is also characterized by having a high level of phenotypic diversity, and three big morphological groups of species can be defined based on vegetative characters. We have simple-leaved Weinmannias, we have these sort of auriculate-leaved plants, and we have these compound imparipinnate leaves. And within each of these groups, we have varying levels of leaf or leaflet sizes and also trichome density or pubescence. So in addition to their scientific interest, they have it, these plants have found to provide multiple uses and ecosystem services, among of which we can mention their use as like wood fuel, timber, but also like the bark is used for leather production due to its high content in tannins. They have been reported to be used as medicinal uses and as melliferous plants. In addition, they are crucial in some ecological restoration projects due to their abundance, but also to their they have this sort of pioneer ecology where they like to be in they have this tolerance for disturbed places. So these are, and in the Americas, deforestation and their use as wood fuel, timber, and bark extraction are threatening the populations of Weinmannia. And for example, out of 49 species evaluated by the IUCN, 16 of them fall under a threat category, including, for example, 3 endangered species. Among of them we can mention Weinmannia costulata, found in Ecuador, for example. So despite all of this importance, very little is known about their evolution and biogeography. However, previous research at the family level have found that Weinmannia from the Neotropics, represented by Weinmannia pinnata in this tree, and Weinmannia from the Mascarene Islands, represented by Weinmannia tinctoria here, form a monophyletic group. This disjunct distribution makes it a very interesting system to study biogeography, as we will see later. In the Neotropics, they are mainly distributed in a diversity of mountain ecosystem forest, mainly through the Andes, but also in Central America and Caribbean islands and Tepuis Formation, Venezuela. But they are also found in other mountainous systems, there's like one endemic species in the Atlantic forest in eastern Brazil, but we also have one endemic species here in the Valdivian temperate rainforest in the south cone of South America. Well, now, so to be able to understand the biogeography and evolution of Weinmannia, we need well-resolved phylogeny. However, very little is known about the phylogeny of Weinmannia itself, and some previous systematics work are mostly family-level studies that include only a few species of Weinmannia and use a limited amount of characters as morphological data, as listed here, traditional Sanger sequencing, angiosperm 353, and more recently our collaborator, Francisco Fajardo, is working on chloroplast sequencing. However, here we propose to use a broader sampling within the genus, species within the genus, and use genome scale data to reconstruct the phylogeny of the group. There are some factors that complicate not only the taxonomy of the group, but also the phylogenetic inference. And perhaps the most important is the pervasive hybridization among coexisting species that was previously reported by some taxonomic studies. For example, here we can see how intermediate morphologies between co-occurring species have been regarded as hybrids in taxonomic studies, and suggesting that hybridization might be common all the way through the range in the neotropics. In addition, we have some taxonomically questionable and broadly distributed taxa that display a high level of phenotypic diversity. One example is here Weinmannia pinnata, that is distributed from southern Mexico all the way through Bolivia to almost like southern Bolivia. And this might indicate that there might be some cryptic species, but also some undescribed species within this range here. Now let's talk a bit about the phylogenetic reconstruction of Weinmannia per se. And for this purpose, our team has gathered more than 1,700 specimens of Weinmannia within the neotropics in countries like Chile, Bolivia, Peru, Ecuador, Colombia. But we have also gathered specimens from Reunion Island, and we have gathered some other specimens of outgroups from New Caledonia and Madagascar. Out of all these specimens, we have sequenced around 900 using the 2B-RAT sequencing approach, which is a reduced genome representation technique that produces fragments of 36 base pairs across the genome of each specimen. So we get thousands of loci per specimen. And so far, we have sampled around 52 species taxa. We have built nucleotide assemblages using iPyRAT pipeline, and the molecular analyses we will show here were done in the structure program to find genetic groups, and to build phylogeny we used maximum likelihood methods implemented in RAXML. And to build our phylogeny, however, we have removed all identified hybrids because of the method we use. We assume bifurcating topologies. So let's just focus on the approach we took in the North Andes to identify hybrids. So we have built a preliminary phylogeny, including all specimens collected in Ecuador and Colombia, which from now on will be called the Northern Andes. And within this preliminary phylogeny, we have found like 4 big clades, represented each one in a different color here. And now let's upon which we conducted structure analysis within each clade. And so let's take a look as an example to the results of this clade 1 right here. So this is a structure analysis plot where each bar represents a specimen, and the colors are basically, these are stacked bars that represent the probability to be assigned to a specific genetic group. And as we can see, for this clade, we found 6 genetic groups. And we identify hybrids as those individuals that have less than 85% of probability to be assigned to a particular genetic group. And as we can see here, indicated in black dots, we have all of our potential hybrids. Following this same approach for all 4 clades within this North Andes, we have identified about 74 out of 201 specimens. That is 37% of all specimens are identified as hybrids in Colombia and Ecuador. So for further phylogenetic analysis, we just remove these ones. And in the case of other regions, such as the Madidi regions, for which we have a much denser sample for each species, we have, well, actually Juan will talk more in detail about the approach we took here to identify hybrids. But for now, I just want to mention that we also found a significant amount of hybrids as shown in this structure plot here. So after several iterations of structural analysis and phylogenetic analysis, here we have our specimen level phylogeny, including 234 specimens of 52 putative species taxa, including our outgroups. And well, the character matrix used for building this phylogeny contains more than 1,000 loci concatenated with a missingness of 31%. Just to mention, yeah, we got like good bootstrap supports, particularly of greater than 80%, particularly for the backbone of the phylogeny. And the tips here are, the name of the tips here are colored by country of origin. Yeah. One of the most interesting patterns we found throughout the phylogeny is that in some cases the specimens that have been assigned to a morphological species don't group into monophyletic groups. And one such example is Weinmannia phagaroides that was found in 3 clades, one in the central Andes, Bolivia, and 2 in the northern Andes in Colombia and Ecuador. So to understand this a bit better, we just went back to our structural analysis, and we just found that, for example, specimens of Weinmannia phagaroides found in this clade right here formed a cohesive genetic group with other specimens that have been assigned to different species taxa, including Weinmannia cundinamarcensis, tomentosa, Weinmannia urtela, for example. And we still need to work on the taxonomy of these specimens. However, at the moment, I have just assigned them arbitrary as a single operational taxonomic unit that I have just assigned as the most likely species, which it might be Cundinamarcensis, say clade 3, genetic group 4, just like a random name. The same thing happened with this other Weinmannia phagaroides found in this other clade that grouped together with other specimens that contained that were assigned to a different names. So this was also the case for some other species as well, such as Weinmannia balbisiana, which we took the same approach. And here we have our same specimen level phylogeny, but with the clades collapsed based on these operational taxonomic units. And just to mention that the ones that are painted in brown here are the ones that contain more than one species taxa, and that we need to investigate more whether these are sort of generating non-monophyletic groups, but we still need to figure out better the taxonomy or to investigate what is going on here. So as I said before, there are multiple factors that complicate the estimation of the phylogeny for the group, and probably the most important is the hybridization, but also we could be facing a scenario where we have ongoing speciation or recent speciation that causes incomplete lineage sorting, and we plan to investigate these using other methods such as coalescence-based methods and phylogenetic networks. In addition, there is a lack of a comprehensive taxonomic treatment throughout the group that help us to improve our taxonomy, or phylogeny. So this highlights the importance of taking a comprehensive approach, integrating molecular systematics and taxonomy. Particularly here, I argue that phylogenetic systematics could help us to have a better taxonomy. At the same time, having a better taxonomy could help us having a better phylogeny, in other words. Yeah. Now let's quickly talk about our results on the biogeographic analysis of the group. And here I will talk about this question we asked regarding the biogeography of the group. And it's about like whether the colonization of the group followed a progression from southern temperate latitudes towards north tropical latitudes through the Andes. And so far, our specimen level phylogeny that I've shown before shows a very strong geographic signal where we have the species that have a more southwards distribution, southern distribution. They tend to group closer to the root of the tree, while the species clades that are more derived tend to come from more northern latitudes. In addition, interesting pattern here is that we have Weinmannia from the Reunion Islands, representing all Weinmannia from the Mascarene Islands, grouping within this big clade containing all neotropical Weinmannia. So I will talk a bit more about that in a few minutes. So using the operational taxonomic units we identified previously, we have built a species level phylogeny, or a species tree, including all one representative of each of the 52 operational taxonomic units. And to better understand the biogeography, we calibrated this species tree in time using 3PL, it's just a program. And for this purpose, we have used as calibration points previously modeled age for the split between Pterophila and Weinmannia, dated to 29 to 34 million years ago. We used a fossil record that was found in the southern central Andes that was dated from 30 to 33 million years ago, and this was placed at the node, the most recent common ancestor of all neotropical Weinmannia, and a fossil pollen record that was found in the North Andes, which was placed on the clay that contains all the North Andes. So these estimates suggest that Weinmannia might have arisen sorry, in the late Eocene, around 34 million years ago, and in the Neotropics, it might have started to diversify around 17.5 million years ago. So just mention that these results are preliminary, and we still need to include some other species, such as the Atlantic forest species, that might help us improve our estimates. As a preliminary test of the northwest dispersal, we have just performed ancestral stage reconstruction for the minimum latitude of each operational taxonomic unit. And in this plot, in this phylogeny here, we have colors represent a gradient going from temperate regions in red towards tropical regions in blue. So we use this ancestral reconstruction to model the node age as a function of the latitude. And as we can see from this regression curve right here, this predictive model already supports our hypothesis of northwards dispersal from temperate northern regions, given that the slope parameter is different than 0. In this case, it's negative, and as we can see from this 95% confidence interval for the estimate and this maximum a posteriori estimate. So in simple words, this suggests that older ancestral species tend to have more temperate latitudes. So to better understand this, here is our first attempt to do a reconstruction of the biogeographic history of the group, and using the dispersal extinction cladogenesis framework, using this epoch model. So applying this approach, we were able to constrain the analysis, ensuring that Weinmannia species from the Mascarenes couldn't predate 10 million years ago, which is the age of the oldest Mascarene island, Mauritius in this case. And This result, in general, supports our hypothesis of northwards dispersal from southern temperate latitudes towards the tropical latitudes in the neotropics. But another interesting pattern we observe here is this suggested long-distance dispersal event going from the neotropics to Mauritius. However, this is something we still need to investigate. Nonetheless, we have good reasons to think that this is possible, given that, for example, in our laboratory, it has been found that hibiscus also show these long-distance dispersal events, as observed by Brock here. But also, I've been talking to Juan the other day, and this type of long-distance dispersal events have also occurred in Lauraceae. So it's not so strange. However, we still need to investigate these, and for the next steps, to understand better the biogeography, we need to expand our sampling into the, well, we are planning to expand our sampling, including species from the Atlantic forest, Costa Rica, southern Mexico, and Caribbean islands. This will help us to improve our biogeographic history reconstruction, in addition to use other approaches as well. We plan to also match divergence times with important geological and climatic change events to understand how these historical events might have affected the biogeography of the group. And yeah, finally, I just want to acknowledge to our broad network of collaborators, and that comes from different institutions in different parts of the world. And I also want to thank you for listening to the talk. And with that, I will pass it on to Juan.
Yeah, okay. So, hello everyone. Thank you for being here and allowed Eduardo and I to share the results. I'm sorry I don't have coffee for everyone. Someday I will say coffee for everyone. But for now, I'm just a postdoc with Sebastian and Christy. I will be close to the microphone so people in Zoom can listen. And I want to talk about this project. The project started as a population ecology project by Alex here. And I talked about this project around a year ago. And we have had a lot of progress on this. Not that we didn't have with Alex. We also keep having progress. And what is interesting is that this project is informing taxonomy. It's informing systematic and evolution like Eduardo is doing. But we also are finding interesting things that will make us rethink the way how we look at hybrids, how we incorporate in ecological and in conservation and even restoration studies. So it's kind of like we want to present here because we want to open the discussion for hybrids into this taxonomical world and beyond that. So before I start, I want to acknowledge all the contribution for all the, I don't know if that works, the contribution of all the collaborators. This is a really amazing team, really diverse team. All the institutions associated with this study, but also to all the local botanists and field assistants and collecting the samples that we are using on this study. So thank you to all of them. So it is easy to agree that hybridization is an evolutionary force that contributes to increase genetic and taxonomical diversity. And in the classical way, how we look at populations that start diverging, and through time start accumulating some genotypic differences or characteristics that make it different through time. One of those is the degree of isolation. But in early stages, that degree of isolation, two distinct populations, or in some cases we can call them species, they can mate and produce fertile offspring. Those individuals are called sometimes fertile, sometimes not. Those are called hybrids. And if those hybrids are successful at all, and they come back close with one of the parental species, we get something called integration, and it's just moving genes from one species to the other and increasing the gene pool of this species. This is the classical way how we see these things. But in current times, we know that history is a little bit more complex, and we have here more reticulated evolution when species can split and can rejoin again. We can have integration not only between sister species, not only between divergent species, but between closer related species in some degree or some species. Let's do it just like that. And that happened in the past, but that could be even happening in the present. What this kind of process does is they result in novel adaptations, moving genes from one species to another, from one branch to the other one. In other words, they don't need to invent the wheel every time. Species can move set of genes that allow them to diversify, to adapt, and not just do it through random mutation. So hybridization is fueling all these important processes that we see. In recent years, people have been paying more attention to hybrids, at least in angiosperm. People working in firms are ahead of us. I said us, angiosperm people, firm people, you are ahead. And then some papers shows, or at least recommended, different ways to incorporate integration and hybridization into the phylogenic systematic work. The classical one is just including or thinking about hybridization, but also plotting different topologies, looking discordant between topologies. This is also similar to this one, showing different topologies. And this is my favorite one, being 3D, showing different connection between branches that happen through time. So it's a different visualization way, because the methods are still relying in bifurcated evolution, like we see in Eduardo's talks. We are still having problems incorporating groups with hybrids. If we think about the neotropics, and again angiosperms, hybridization has been thought that is uncommon, that is rare. Mostly because of three reasons: reproductive specialization in angiosperms, narrow species distribution, and an unrelated species share the same niche. However, in a species with reproductive semipermeable boundaries, hybridization can occur, and when the hybridization happens and is successful, it can even produce something called hybrid zones. Hybrid zones are really interesting things because we have three main hybrid zones. One is called the tension zone, where hybrids have lower fitness than the parental species. So they can occur, they occur in this kind of Kleiman transition between the species, but they don't survive. They are just there. Through this talk, I'm going to represent hybrids with black dots. Another hybrid zone is the bounded superiority zone. Bounded superiority zone, in which hybrids have higher firmness than the parental species. They can occupy different environments. They can even bud cross, and now bud cross are represented in red through the whole talk. And this is the one that people like to find, like to see, because this one is the one that leads to speciation through hybridization. Species get hybrids. Hybrids have a higher fitness, and voila, we get a new species. But there's another one, a really interesting one, that is called the mosaic zone. In this hybrid zone, hybrids and bud cross individuals can have higher fitness in some places, but lower fitness in different ones. And in this one, they occur within the species boundaries. This case produce gene flow between the species. Not just like a speciation or they are dying out. they can keep and maintain the gene flow between the species. And this is a really interesting hybrid. So let's talk now about Weinmannia and why we are here talking about hybridization. So in Weinmannia, species often overlap in distribution. They like to grow in poor soils, like this seedling, and like living this kind of disturb, like in a Weinmannia, they like drama. They don't like to live in a nice place. They like, crazy places. Pollen is dispersed by insects, just generalist interactions, and get attracted by polliniferous discs, nectariferous discs. Attracting pollinators, they just go there, and then they have these really tiny seeds, and Eduardo mentioned, and they are dispersed by the wind, so there are no intermediation from frugivores. So they just get generalistic pollen, pollinators, seeds just go flying around, trying to find a drama place to live, and they overlap there. So it's a very interesting group when species can coexist. Species are differentiated by leaf traits, like Eduardo mentioned, imparipinnated versus simple, the margin morphology, and in some cases, the leaves are glabrous or not. So very simple taxonomy here. But in Bolivia, at least in Bolivia, to the whole range, this happened, but in Bolivia, intermediate morphologies are often found, and they are described or they are identified as hybrids, especially in places when you can assign two parental species and coexist in the same locality. So you get an intermediate morphology with two things that looks like you are a hybrid, and probably you are a hybrid from these two parental species. Only Mariri, now we are just like narrowing just to Mariri National Park, we have at least 20 species of Weinmannia current in geography. And in the elevational range from 1,500 meters to about 4,000 meters in elevation. We have good data for 13 of those 20 species. I'm going to focus on those 13 species for now. And this is the distributional, the elevational range distribution. When seen that all the species overlap in elevation, even the species that has narrow distribution, they overlap with other ones. And they also overlap in geography. This is the region that we are focusing now. So with all this, how you say that, melting pot and this nice situation, something like that, when we have a family that has intermediate morphologies, species overlap, it looks that they don't have any specialization, and they overlap in, I won't say the niche, but in geography and in elevation. So we decide to ask three simple questions. We are just like exploring hybridization here. And for these 13 species that we found in Bolivia, we just ask if when we identify, I'm going to read them better. Individuals identify a species in the field based on taxonomical keys from genetic clusters. So what we see as a species is genetically related. The second question is, do the individuals with sign of hybridization, so these intermediate morphologies correspond to F1 hybrids with bar cross individuals? So just in some sort of integration. And what do the hybrids occur in relationship with the parental species in the range in geography and elevation? So for this, we use 409 individuals collected in the permanent and temporary plots in the Mariri project. This is a really good thing, so we have those plots. And we added some samples with elevational transects. We prepared the two-wheel library, and it's one of these sets of next gene sequencing that we can perform here in Christy Edwards' lab. We use for the first question just structure to explore the genetic clusters. We estimate hybrid index in genotype to estimate how much a hybrid is related to one of the parental species, and then we just compare the elevational and geographical ranges. And don't worry, I'm going to stop and explain everything at some point. So for the first question, what we see with taxonomy and what we identify are date related, and then we use a structural analysis. And Eduardo did a really good explanation, but let me refresh what he said. So we define hybrids as an unmixed descendants of two or more distinct ancestral populations. So when two things are diverging, they interbreed, we call them hybrids. And what the structure does, it estimates the number of ancestral populations, or what we call genetic clusters, and the proportion of admixture assigned to an ancestral population from each individual. So in this case, for instance, it's assigned to this kind of, what is that, gray color. This one is this salmon color, blue color. There are three distinct populations, and this individual is an unmixed with a small proportion of blue, a little bit more of gray, and then a little bit more of blue. And what we did, based on those proportions, we choose 85% of probability of proportion assigned to a specific group. If you are below, you are called hybrid. If you are above, you are assigned to one of those groups as a species. And this is how our data looks for the 409 individuals. Not as nice as the previous one, but way more interesting. So we found 15 genetic clusters or ancestral population, calling it genetic cluster now, and they correspond to 12 morphological species. This is a really crazy slide, so, okay. So let me walk you through the results. So we have here a sample of 4 of the species. Based on morphology, botanists, I say we, but it's the team, it's not me. That was technically Alfredo Fuentes and his people. They identified 311 individuals as a species based on morphology. But when we run the structural analysis, we found 286 individuals were identified as a species. That means 80% of the time they were right assigning a name, and that name corresponds to a specific group, like in this case. But they identified 98 individuals based on morphology and, as a hybrid, and we found 123 as hybrid. So they underestimate the number of hybrids. And you can see that in the plot here. Some individuals are mixed individuals, and they are assigned to some of the specific groups. So that means that in Bolivia, taxonomists are really good in identifying the species. However, we are still lacking the tools, not the knowledge, I think that the tools, for identifying the hybrids better. So something that we need to keep in mind. Two of the interesting results that we have here is the case of Weinmannia L and Davidsoni. Individuals identified as these two taxonomical names, they form one genetic cluster. So two things could be happening here. One, they are misidentified as individuals and all of them belong to the same species. Or the second one is that Davidsoni is a synonym of Nebularum. And in this case, Davidsoni is a species that was recently described, and the difference with Nebularum are slightly larger leaves in Davidsoni. So it's a really kind of a small or weak character for such a crazy group that vary that much in morphology. So really interesting result. But we also get something that is kind of the opposite than this. We get a group called Weinmannia reticulata, when we find 6 individuals that belong to a different genetic cluster. We call it, I'm calling it reticulata 2. Really, I'm a really creative person. So, what is interesting is that the morphology of these two groups is almost identical. The main difference is that these 6 individuals, at least in 5 of those 6 individuals, leaflets are described as being concave. So it's a really tiny character, but it looks that there's something interesting here. So it could be cryptic speciation, could be something funky happening with the genome of these 6 species, but it's something really neat that we just need to explore. Then move to the second question. So what we look, or what we define as hybrid, are they real hybrids, F1 bud cross individuals? And from this one, we go back to the structure, and we found that 123 of those 409 individuals are hybrids. So they have proportions smaller than 85% assigned to each of the groups. Here we have a really interesting table when we can see all the pairwise interactions. We have 31 hybrid combinations in this group, and technically all the species hybridize. This is a better way to illustrate that. I like that one better. I like craziness sometimes. And here we see all the species hybridize at least once. Interesting, reticulata, reticulata 2, they only hybridize among them. But except for that pair of species, every single species hybridize. And here, bars represent the number of, a proportional number of hybrids. So that shows that hybridization is dominated by these four species: Nebularum, Crassifolia, Jungacensis, and Fagaroide. We can see that also in the table. Still, everyone hybridized in this group. So, I mentioned the hybrid index. That is, just estimate how similar each hybrid is to each of the parental species. So here, if you are around 0.5, we call them F1. That means you have equal parental contribution for both of the species. And if you are going towards 0, that means that you are closer, genetically closer to the species or the parental species A. And if you are moving from 0.5 to 1, you are closer to the parental species B. And that will suggest some sort of introgression. And this is our data. So 56% of the time we get F1s, and 44% of the time we get bar-cross individuals. Bar-cross individuals. Really interesting, showing that we really have gene flow. It's not just hybridization dying out. I like this table. I don't know, I don't like tables, it's just sarcasm. By this table, at least I can illustrate that 68% of the combinations, the hybrid combination, they have both F1s and bar-cross. We only have four cases when we only have F1s. That could be due to sampling, that could be due to many things. But at least focusing in these results, we have F1 of bar-cross individuals in 68%. But looking at the bar-cross individuals by itself, we even have more bar-cross individuals than both combinations. So in 87% of the combination, we get bar-cross individuals. To illustrate this better, let's go to the third question. So where do the hybrids occur in relationship with the parental species ranges? So here, looking at the first combination, hybrid combination, we are going to look at the geographical and the elevational distribution. Here we have latitude, longitude, west to east, south to north. And here we have elevation for the parental species 1 and 2, and then hybrid index. So how much or how related or how much genetic material you have from each of the species. And again, black dots, hybrids, red bar-cross. So here we can clearly see that when the two species occur, you get hybrid and bar-cross individuals. And you look at the elevation, when the species overlap, it's mostly when you get a lot of the bar-cross and the hybridization individuals. What is interesting here is that bar-cross is not going in only one direction, it's going in both directions. That means that it's a gene flow between the two species. So you are getting, I don't know, more a community, everybody helping each other. Maybe they are not helping, I won't take that one off, but they are sharing the things. And here is, if we look at the samples that we have. So this is a sample of Crassifolia with simple leaves. This is a Jungacensis with compound leaves. And this is a hybrid. And I don't know how to differentiate that one. But someday we will know. just give me more funding and we will find out. And then this will suggest that we have a mosaic hybrid zone when both species can coexist with hybrid and bud crossing individuals within the parental distribution of the parental species. This is another case with Phagaroides and Crassifolia. Same thing. When the species overlap in distribution, you get bud cross and hybrids. But what is interesting in this one is, in this case, Phagaroides and Crassifolia don't overlap in elevation and ranging. But still, we get hybrids and bud cross individuals again in both directions. Here, we have more image, more picture for samples. We have, again, Crassifolia, simple leaves, Phagaroides, compound leaves. Phagaroides is one of the species that Eduardo was showing, and it's not in the same group. So, I won't say monophyletic, but we have problem with the taxonomy of this group. And then one possible reason is like this kind of thing. So here we have this individual correspond to this bud cross individual here with Crassifolia, closely related to Crassifolia, and here it has simple leaves. You can think that they look like. Here we have another one, that is this one, just 0.62, so just a little bit further than the limit that we arbitrarily assign for being hybrid. But this one has imparipinnated leaves. No as imparipinnated as the original Phagaroides, but they are not simple leaf. And then we have this hybrid here, an F1, 0.5 proportion, that has also imparipinnated leaves. See here, it has two small leaflets. So this graph, all this information shows the system that we are living with. We have species and we have a bunch of hybrids that can look like, but sometimes they don't look like, and they start combining everything. So it's a really interesting group to explore these morphological and developmental questions. Again, this one also shows, also just a mosaic hybrid distribution, gene flow between species, no speciation. And I mentioned conservation and ecology at the beginning of this talk, and I haven't really talked about that. So I want to use this example for that. So we just look at Phagaroides, Weinmannia Phagaroides, and this is 6 of the interactions that we have from then. Phagaroides is the species with higher elevation distribution. It hybridizes or produces bud cross individuals, at least in the sampling that we have with these 6 species. But what is kind of interesting for me is that hybrids and bud cross, no matter how related you are to the species, they tend to occur in the elevation of Phagaroides. Same happen here, same happen here, same happen here, and same happen here. This is the only 2 that are not exactly in Phagaroides, and this individual here. But I would think that this could be a potential to study if there is an advantage of hybridizing with Phagaroides. And in this world where we have to think about climate change and this kind of situations that we are living on, this is a really good system to start looking at the effect of hybridization, and maybe a rescue hypothesis. Maybe hybrids are getting hybrid with Phagaroides in lower elevation are getting selected against, but in higher elevation they are selecting they are favored on selection. And again, this is a really interesting family because it's a pioneer family who like to live in a really disturbed, technically you get a landslide. You get Weinmannias and then you get Cecropias. And this is the species that start getting first there. So it's a really good system to also think about restoration. So incorporate restoration, conservation, and ecology through hybridization. So as kind of invitation is to stop thinking that hybrids are negative that should be selected against, and see them more as an opportunity to expand our knowledge in multiple ways. So So conclusion, we can say that taxonomical groups form genetic clusters. So at least in Bolivia, taxonomy had been developing these keys or these skills to identify the species. And although all the species produce hybrids, they maintain gene flow, we can still see molecular and morphological differentiation. Even when we see these intermediate individuals, we are still able to define to identify the species. And hybrids seem to occur in mosaic hybrid zones, promoting genetic flow in between the species more than speciation through hybridization. So again, kind of circulating genes that maybe are helping them to adapt, and again, adapting in these climate change scenarios that we are living on. So what is next for us? This is the phylogeny developed by, built by Eduardo. And we have most of the species in the central Andes. We are lacking some of them for the hybridization studies. Here we can, at least from this one, we can see that it doesn't matter the phylogenetical relationship, you still hybridize. So we want to expand this study to the northern region, to Ecuador and Colombia, and some samples that we have from Peru. Let's start exploring if hybridization is local or they can even hybridize. We can see signs of hybridization among the different depths in the phylogeny. That's a paper that someday I will have, and then I will retire after that one. It's going to be called How Deep Is Your Love? So we are going to look at how deep this relationship can happen and still hybridize. So we are going to also push advancing taxonomy, generating keys that include hybrids. So we need to improve the ability to identify the hybrid when it's possible. Sometimes no, but if we can improve that 80% threshold will be nice. And also expand to ecological studies. Hopefully not only in the lab, hopefully also doing some reintroduction or moving things. So I need to think more about that. That's why we need more time and funding. And then with this, I think that I open the floor for questions to Eduardo and I.